A-level biology hacks with Dr. Biology. So this is experimental data. So how do you evaluate correlations? Well, correlations come in different formats. So usually it's a positive correlation, negative correlation, or no correlation at all. Usually the questions they give you are related to evaluating some information. So some data that they've given you, and you've got to determine, is it correlated? Does it show evidence for a particular thing, or is it evidence against? So usually the evidence for something is that usually it's a large sample. You would need to describe the type of correlation, whether it's positive, negative, or there's no correlation in the data. The evidence against, usually these are the main points that come up time after time. So correlation, but it does not mean a causal effect. So it might show a relationship, but it does not mean a causal effect. Is there a wide range of points that overlap? And we'll have a look at an example in a minute. Usually other named factors are involved, so it could be named abiotic or biotic factors, and variation in organisms being measured at a particular abiotic factor. Let's look at some examples. So here we have a graph showing the sample of patients with Huntington's disease, and it shows the number of CAG repeats, and it shows some information, and it says people can be tested. It says use information in the graph to evaluate this suggestion. So remember, evaluate, you need to show the pros and cons or evidence for, evidence against. So first of all, you need to describe the graph. So definitely it is a negative correlation. However, there is a very wide range of data points at each number of CAG repeat. And you can see actually with a different number of CAG repeats, there's many overlaps. Therefore, that would suggest other factors may be involved in the age of onset. This separate one, this is an ecology-based one about water beetles in 30 lakes. So you can see lakes without fish, lakes with fish, a very loose correlation there. And it said a student concluded that a decrease in acidity caused an increase in the number of water beetle species. So a decrease led to an increase of water beetle species. Evaluate this conclusion. Well, let's talk about the correlation. So I would say there was a weak positive correlation, but you do not get a mark for that because it's pretty weak. So again, correlation, but it does not mean that's a causal effect. There could be other abiotic or biotic factors. So, for example, due to the presence of fish, there might be nutrients, for example. You do need to say what the other factors would be. You need to qualify your answer. There is a variation in the number of beetle species at the same or similar particular pH. So you can see there's a massive variation there, even at 1 pH. For example, you look at pH 5.5 and the number of water beetles, there's a large amount of variation. The results are very spread out again, but the positive is that they looked at 30 lakes, which is a large sample. This is about the relationships between the percentage of fat in the diet and the death rate from breast cancer in 24 different countries. So you can see death rate from breast cancer and then percentage of fat in the diet. The question asks, some people have used the graph to conclude that a high percentage of fat in the diet causes breast cancer. Evaluate. Well, again, it is a positive correlation. You can say that. But as we know, a correlation does not always show causation. So it may not be the percentage of fat in the diet may not be the causal effect. There may be some other effect, e.g. age, or it could be something like amount of exercise. You need to really kind of make that up. The evidence against the positive correlation, though, is you can see at the same percentage of fat, you've got different death rates. So, for example, a really specific one, if you look at 35, you can see that death rates vary all the way from 800,000 death rate from breast cancer per 100,000, 8 per 100,000 compared to 20 per 100,000. So that's a very big difference. Also, some countries with higher death rates, in fact, have lower fat intake. So you can see the ones that are above the line. There you go. So there's a really good example. Here's my last example. So this is looking at urine and blood samples from a girl with diabetes at intervals over a 1-year period. So blood glucose concentration. Urine glucose concentration as color value. That is a subjective measure. Okay, it's not a quantitative, it's a qualitative measure. It says use the graph to evaluate the use of the urine test as a measure of blood glucose concentration. Well, there is a very, very loose positive correlation. It is directly proportional, but you can see the vast range of results for a particular value, and there is many overlaps in the data between the different urine glucose concentrations. And the urine test is an arbitrary scale, so meaning it is subjective. It's not directly related to concentration. And basically you've got a few color values. So there we go. Okay, so I hope you have found that useful. There's more A-level biology hacks to come. Please do subscribe, and I'll see you soon.